Assalamu alaikum, dear students. In today's lecture, we will talk about a disease known as schistosomiasis, which is caused by a different schistosoma species. Chapter number 16. Outlines of today's lecture are: we will discuss the disease schistosomiasis in detail, where we will discuss introduction, then history of schistosomiasis, epidemiology of schistosomiasis, life cycle of schistosoma, pathogenesis and treatment and preventions. Lecture outcome. So after watching and listening to this lecture, a student will know about the various aspects of this well-known disease, schistosomiasis. Let's start from the taxonomic position. So, superfamily Schistosomatidae. Flukes of this family, superfamily Schistosomatidae, they are peculiar in a way because they don't have second intermediate host in their life cycles. And another way, they are unique because they mature in the blood vascular system of their definitive hosts. The flukes are parasites of fishes, of turtles, of birds and mammals throughout the world. Several species are parasites of humans and causing misery and deaths wherever they are distributed in the world. Most species are dioecious. Several species are parasites of human beings and they cause misery and deaths wherever they are distributed in the world. The families Sanguinikulidae and Spirochaetidae, they parasitize fishes and turtles. The family Schistosomatidae, however, includes species that are among the most dreaded parasites of human being. Dreaded mean that they are fearful or feared parasite of human beings. To date, there are 12 species of schistosomes have been reported in Africa. Taxonomy of this group and validity of some species of parasites have been subjects of controversy for years, and final decision on species recognition awaits further studies on this group. Family Schistosomatidae. Schistosoma species and the disease or the condition known as schistosomiasis. Schistosomiasis is a major human disease with 600 million people and 74 countries at risk. Some 200 million people are infected with schistosomes, of whom 120 million are symptomatic and 20 million suffer from severe disease. History of the schistosome. 3 species of schistosomes are of vast medical significance, which are: number 1, Schistosoma haematobium, Schistosoma mansoni, and Schistosoma japonicum. All are parasites of humans since antiquity. Bloody urine was a well-recognized disease symptom in northern Africa in ancient time, which was caused by this Schistosoma haematobium. At least 50 references to this condition, schistosomiasis, have been found in surviving Egyptian papyri, and calcified eggs of Schistosoma haematobium have been found in Egyptian mummies dating back to 1200 BC. So from this you can imagine how old or ancient disease this is. Hulls presented a well-reasoned hypothesis that uh, the curse that Joshua tribe placed on Jericho tribe can be explained by an introduction of like Schistosoma haematobium into the communal will by the invaders. These were two tribes, Joshua and Jericho, and they always were fighting with each other. So what happened? Like one of them, they what they did, they uh, introduced deliberately the eggs of Schistosoma haematobium into their communal well, to the communal well of the Jerichos. So what actually Joshua did, uh, the Schistosoma haematobium eggs are found in human fecal material. And what they did, they introduced it into the communal will of the Jerichos. And Jericho, they got ill, they become ill with the schistosomiasis, and got their revenge from Jericho. So then by removal of the curse occurred, how it occurred after abandonment of the Jericho and subsequent drought eliminated the snail host, uh, Bolanus truncatus. So that's how this curse then eliminated. So today there is Jericho area, Jordan, is well known for its fertile land and healthy, well-nourished people. The first Europeans to record contact with Schistosoma haematobium were surgeons with Napoleon Army in Egypt, 1799 to 1801. They reported that hematuria, called bloody urine, was prevalent among the troops, although the cause of course was unknown. So nothing further was learned about schistosomiasis or hematobia for more than 50 years until a young German parasitologist, Theodor Bilhars, discovered the worm that caused it. So this Theodor Bilhars discovered this worm, and this is why this disease is also famous with the name of bilharziasis. So he announced his discovery in letters to his former teacher, von Siebold, naming the parasite Destomum haematobium. But actually it was not Destomum haematobium, it was Schistosoma haematobium. Later on the Destomum was replaced by Schistosoma. Bilhars died of typhus at the age of 30. So again the work stopped. So during the next few years, it was discovered that 30 to 40% of the population in Egypt bore infection of Schistosoma haematobium, and the worm was even found in an ape dying in London. So the peculiar morphology of the worm made it clear that it could not be included in the genus Destomum. So in 1858, Vineland proposed the name Schistosoma. 3 months later, Cobbold named it Bilharzia after its discoverer, the Bilhars. This later name became widely accepted throughout the world, and the parasite was even given the nickname Bilharis by the British soldiers serving in Europe during World War I. Today, however, the strict rules of zoological nomenclature decree the schistosoma has priority, and is thus the current name of the parasite is Schistosoma. Even so, health officers in many parts of the world, they showed sign next to ponds and streams that warn prospective bathers of the danger of bilharzia. Nonetheless, schistosoma is a fitting name referring to the split body because of the gynocorporal canal of the male. We'll discuss this later in the later slides. What is gynecophorial canal and why it is present in male and what is its function. While information was accumulating on biology of Schistosoma haematobium, some investigators began to doubt whether it was a single species or whether two or more species were being confused. So the problem was confounded by the observation in some patients of eggs with terminal spines in both urine and feces. So whenever eggs with lateral spines were noticed, they were ignored as abnormal. In 1905, Sir Patrick Manson decided that the intestinal and vesicular urinary bladder schistosomiasis usually were distinct diseases caused by distinct species of worms. He reached his conclusion when he examined a man from the West Indies who had never been to Africa and who passed literally spined eggs in his feces, but not at all in his urine. So Sambon argued in favor of the two species concept in 1907, and he named the parasite producing literally spined egg Schistosoma mansoni. Because actually in the start they knew it is Schistosoma haematobium, but later when they were also discovered in the urinary bladders or in the urine, and at the start it was actually like discovered in the urine. So it was discovered in the fecal material also. So then there was this agreement between the scientists. Some were like accepting one species, or some were like not agreed with this idea, and they were of the idea that these are two separate species. But like someone in 1907, he clearly mentioned it, that these are different, and he named it Schistosoma mansoni. Japanese zoologists had already detected still another species by this time, but their reports were generally unknown to Europeans, and that was later on named as Schistosoma japonicum. However, the eminent German parasitologist Loss disagreed and brought the full sway of his reputation and dialect against the notion, and even stated that he had seen a female worm with both kinds of eggs in its uterus. But Sambon, undaunted, replied that until Professor Loss could show me an actual specimen, I am bound to place the worm capable of producing the two kinds of eggs with the phoenix, the chimera, and other mythical monsters. So then there was this disagreement that it continued, because the Loss, he was like agreed that I have seen both of kinds of eggs in one female. But Sambon, he replied that no, it's not possible. You have to show me the actual specimen. Otherwise, I will say that what you are talking, that there were two kinds of eggs in the same female. It is just like someone say that I have seen a phoenix and I have seen a chimera or other mythical monsters. So the question was finally resolved by Lipper in 1915, and he first visited Japan to acquaint himself with the work of Maeri and Suzuki on Schistosoma japonicum. Then working in Egypt, he discovered that Cercaria emerging from the snail Bolanus species could infect the vesicular veins of various mammals, and they always produce eggs with terminal spines. So those emerging from a different snail Bionfilaria species infected intestinal vein and produced literally spined eggs. So it was soon determined that Schistosoma mansoni had a broad distribution in the world, having been widely scattered by the slave trade. and it is now widespread in Africa and the Middle East, and is the only blood fluke of humans in the New World, with the possible exception of a small focus of Schistosoma haematobium in Suriname. The original endemic area of Schistosoma mansoni was probably the Great Lake region of the Central Africa. Suriname is a small country in South America. So, while Cobalt, Vineland, Bancroft, Sembon, and other, they were like wrestling with each other with the problem of Schistosoma haematobium and Schistosoma mansoni. Japanese researchers were investigating a similar disease in their country. Uh, for years, physicians in the province of Hiroshima, Saga, Yamaguchi, had recognized an endemic disease which they characterized by an enlarged liver and spleen, ascites, and diarrhea. So, at autopsy, they noted eggs of an unknown helminth in various organs, especially in the liver. In 1904, Professor Katsurada of Okaima recognized that larvae in these eggs resembled those of the Schistosoma haematobium. So, because he was unable to make a post-mortem examination of an infected person, he started using or examining local dogs and cats in hopes that they were reservoir for the parasite. He soon found adult worms containing eggs identical to those from humans and named them Schistosoma japonicum. The experimental elucidation of the life cycle of various Japanese researcher was a milestone in the history of parasitology and formed the basis for Lepper's work on blood flukes in Egypt. The distribution of Schistosoma japonicum is limited to Japan, China, Taiwan, and Philippines, and Southeast Asia. In most recent years, other species of Schistosoma parasitic in human have been distinguished, and Schistosoma species seem to be evolving actively. Next, we will talk about the morphology of Schistosoma. Although Schistosoma species are generally similar, they are similar structurally or similar in their structure, several differences in detail are enlisted in table say 16.1. I will show you that table in the next slide. Considerable sexual dimorphism exists in the genus, male being shorter and stouter than males, and they are muscular. I will show you the structure in 16.5. The male have a ventral longitudinal groove. As I told you, we will talk about that later. So this is what gynecophoral canal is. It is present in males. It's a ventral longitudinal groove present in males, where the female may normally reside. Because generally the female has weak musculature and the male have strong musculature, so that's why the female reside inside that gynecophoral canal. The mouth is surrounded by a strong oral sucker, and the acetabulum is near the interior end. I will show you in the figure 16.6. There is no pharynx. Here in this structure you can see the gynecophoral canal I was talking about. So this is the male schistosome, and this is the female schistosome, which is living inside this gynecophoral canal. So it's a ventral groove, ventrally it's found ventrally on the male side, and the female these females they live inside in this gynecophoral canal. So this is male with these strong suckers and with strong body musculature. This is the female with weak body musculature, and this female, it lives inside the ventral groove or the gynecophoral canal of the male. This is the table characteristics, and these are the three different species and how you will differentiate among them. So Schistosoma haematobium, Schistosoma mansoni, and Schistosoma japonicum. Tegument papillae. Now here these tegumental papillae are small tubercles. Here large papillae with spines, and they are smooth. Size: male. Length is from 10 to 15 millimeter. 10 to 15 millimeter. Here 12 to 20 millimeter. Width is from 0.8 to 1 millimeter. 0.8 to 1 millimeter, and in case of japonicum 0.5 to 0.55 millimeter. Female length. So in case of haematobium, it is 20 millimeter, and Mansoni it is 20 millimeter, and japonicum it is 26 millimeter. Width is 0.25 millimeter, 0.25 millimeter, and 0.3 millimeter. Now number of testes. So in Schistosoma haematobium there are 4 to 5 testes, and Mansoni there are 6 to 9 testes, and in japonicum there are 7 testes. Position of ovary: near mid body and anterior half posterior to mid body. Uterus with 20 to 100 eggs at this at one time, average 50. And Mansoni, few follicle, short, uterus short, few eggs at one time. Uterus is long, may contain up to 300 eggs, average 50. Vitelliria. So few follicles posterior to ovary, few follicles posterior to ovary, and in case of japonicum, and in lateral fields, posterior quarter of the body. Here then we talk about the eggs. So in case of Schistosoma haematobium is elliptical with the sharp terminal spine, 112 to 170 micron meter, cross 40 to 70 micron meter. Elliptical with sharp lateral spines, 114 to 175 micron meter into 45 to 70 micron meter. Oval to almost spherical, rudimentary, laterally, and 70 to 100 micron meter into okay, so rudimentary lateral spines, 70 to 100 micron meters into 50 to 70 micron meters. This is the difference on the basis of different structures and morphology. The paired and other characteristics of the Schistosoma species. So the paired intestinal ceca converge and fuse at about the midpoint of the worm, and then continues a single gut to the posterior end. A male possesses 5 to 9 testes, and as we discussed in the table, according to species the number varies according to species. Each of which has a delicate vas efferens. These vas efferences, these ducts then combine to form a vas deferens, and this vas deferens then dilate and become a seminal vesicle, which opens ventrally through the genital pore immediately behind the ventral sucker. So behind the ventral sucker, this open to the genital pore. I will show you in the diagram. The serous pouch, serous and prostate glands are absent. The suckers of females are smaller and not so muscular as compared to those of males. The ovary is anterior or posterior to or at the middle of the body, and the uterus is correspondingly short or long depending on the species. So we also discussed this in the table of morphological characteristic table. Then biology. Adult worms live in veins. They drain certain organs of their host abdomen, and the three main species have distinct preferences. How? Like for example, Schistosoma haematobium lives principally in veins of the urinary bladder or and plexus. Schistosoma mansoni prefers the portal veins draining the large intestine, and Schistosoma japonicum is more concentrated in veins of the small intestine. So these all different species have like different preferences to live in. Schistosoma haematobium is found in the urinary bladder plexus. Mansoni is found in the large intestine, portal veins of the large intestine, and japonicum is in the veins of the small intestine. Female worms are usually in the gynecoporal canal of male worms, where copulation takes place, and there are other physiological reasons for this location. Meanwhile, the female live in the gynecoporal canal. So, as I told you, that female have weak musculature and weak oral sucker, and they need the help of the females because male are like strong, have strong musculature and strong oral suckers. So the female live inside the gynecoporal canal of the male for safety, for attachment, and for movement in the veins. The more robust muscle of the male allows the paired worm to work their way upstream into smaller veins where the female deposit eggs. So, like female cannot go upward. It is with the strong muscle, the robust mean the strong muscles of the male, which allow their paired struggle upstream into the veins, smaller veins, and where the female will deposit its eggs. The eggs must then cross the wall of the venule, some intervening tissue, and gut or bladder mucosa before they are in a position to be expelled from the host. So the mechanism by which this escape is achieved is not self-evident and has been the subject of much speculation. Spines on the eggs were traditionally credited with contributing to the expulsion, but this feat is also accomplished by S. japonicum and other Asian schistosomes that have only the most rudimentary spines. It appears that the worms enlist the egg of their host. According to File, the endothelial cells lining the venule actually move over the schistosome eggs to exclude them from the lumen in figure 16.9. These are this is the scanning electron micrograph of endothelial cells in eggs of Schistosoma japonicum in vitro. So here you can see these are the eggs which are newly hatched, newly given or expelled by the female worm, and the endothelial cells are moving over them. So like you can see, the endothelial cells are already moved upon them, and these are ready to move on these eggs, this one egg too. Damian and Doinoff et al. Postulated that the worm then exploits the host immune response to transport its eggs to the lumen of the gut or the bladder. So the erupted egg stimulate a granuloma to form around it in figure 16:15. The granuloma, consisting of motile cells such as eosinophils, plasma cells, and macrophages, then moves to the intestinal or a bladder lumen carrying the eggs with it. Once in the lumen, cells of the granuloma disperse and the egg is excreted with the feces or urine. This is the structure of the granuloma formation. So here you can see eggs of Schistosoma mansoni and the granuloma and intestinal wall. Here you can see this is the egg. Okay, these are the eggs, and this M is for mucosa. These are the mucosal cells, and you can see here the infiltration of the various leukocytes, different macrophages and eosinophils. They infiltrated in this area. They can be seen here. So in any case, about two-thirds of the eggs do not make it, and large numbers built up in the gut or bladder wall, particularly in chronic cases in which the wall is toughened by a great amount of fibrous tissue. So of course many eggs are never expelled from the venues but are swept away by the blood, eventually to lodge in liver or capillary beds of other organs. By the time eggs reach the outside by way of urine or feces, they are completely embryonated and hatch when exposed to the lower osmolarity of fresh water. The mechanism of hatching is poorly understood. We will continue this lecture. Keep in touch. Reference Foundation of Astrology edition by Gerard Smith. Thank you so much for watching and listening.